In this video, I'm going to go through the autonomic nervous system, which is the end of the Unit 6 notes. So it starts on page 9, and at first, what I'm doing here is just describing the autonomic nervous system. So I'm going to switch over to some pictures to just try to situate the autonomic nervous system with the rest of what we've talked about for you. So the autonomic division of the nervous system, it's part of the peripheral nervous system, although there are components in the central nervous system too. But basically, what we're focused on in this unit is motor control of effectors other than skeletal muscle. So in other words, nervous control of smooth muscle, of the walls of blood vessels, of glands, of the GI tract, of the respiratory tract, urinary, reproductive tracts, sweat glands, endocrine glands. So that's what we're focused on here with the autonomic nervous system. So the real big difference is that the effector is different. And I know I am talking about the autonomic nervous system in context to the peripheral nervous system. And just to take a look at another picture that we've talked about this is from our book. It's a little bit busy, but basically all they're trying to show us here is like in blue, that sensory information comes into the brain and spinal cord. We make decisions about that information, and then in red, we take action on those divisions. And so very broadly, the autonomic nervous system, some people, some authors will talk about sensory neurons being part of autonomic. We're not going to look at that. They also talk about parts of the central nervous system being part of the autonomic nervous system. And although I'm not going to directly look at that, I'm just going to mention, remember how the hypothalamus has a lot to do with the autonomic nervous system. So the hypothalamus and the brain stem receive sensory information, and decisions are made to take action in involuntary subconscious effectors like the heart and blood vessels and the GI tract and so on. And so definitely the central nervous system has components, especially hypothalamus and brainstem, that are controlling the autonomic nervous system, and therefore, they could be considered part of the autonomic nervous system. But really, autonomic nervous system, what we're going to talk about today is mainly this motor output. So when we send a motor command out to the body, it can go to a voluntary source. There, we're talking about voluntary, like skeletal muscle, right? So the somatic nervous system controls skeletal muscle, and we talked all about that in Unit 4. Okay, so I'm not going to go back over that. And that's the neuromuscular junction that I'm referring to. But other than that, we can also control smooth muscle, the heart, glands, like I was saying, and that's the autonomic nervous system. And the autonomic nervous system is broken into two subunits, the sympathetic division and the parasympathetic division. Okay, so we're primarily focused on, this motor output to autonomic effectors in this unit. So I'm going to take a look at another picture from our book. Here what they're doing is they're comparing parasympathetic to sympathetic. So those are the two divisions of the autonomic nervous system. So these two divisions, taking a quick look at this side-by-side comparison, when I look at the effectors that are innervated by the parasympathetic division, okay, the eye, salivary glands, heart, lungs, etc. I won't read down the list. You can compare this for yourself. And then I look at the sympathetic list, okay, the eye, the salivary glands, there's the skin, that's unique, the heart, the lungs. What I want you to notice here is that by and large, the effectors are the same. So in other words, it's not the case that the parasympathetic division is going to innervate like half of the autonomic organs and then the sympathetic innervates the other half. Both sympathetic and parasympathetic divisions, by and large, innervate all of the involuntary organs and have different effects. Okay, so they're working in opposition. They're working antagonistically. And so that's the basic premise of the two divisions of the autonomic nervous system. that both divisions are innervating the same structures and just having the different effects. There are exceptions to that. And so there are structures, and a good example is the smooth muscle and the walls of blood vessels that are only innervated by one division. And that's the sympathetic division. We'll talk about that in AP2, so I'm not really even going to talk about that in this unit too much, but I'm just giving this example that there are some areas, and like the skin is another example. By skin, they mean like the sweat glands and the erector pili muscles are innervated by the sympathetic division. They're not innervated by the parasympathetic division. And so increased or decreased input by the sympathetic division can ramp up or slow down those effectors that are only innervated by sympathetic. And for the effectors that are innervated by both parasympathetic and sympathetic, um, what's happening is there's a baseline um, activity of both that are in a balancing act. It's not the case that, you're either innervating the heart with just parasympathetic and no sympathetic, or vice versa, that, we're all sympathetic or and no parasympathetic. There's a balancing act going on in all of these involuntary structures where they're being innervated by both divisions. And these divisions are really, if I wanted to put one like summary word on this, are responsible for homeostasis. They're responsible for monitoring what's going on in the body and keeping the lungs, the heart, the stomach, our blood pressure, and so on within the normal range. Okay, and so there's this, balancing act that's going on between sympathetic and parasympathetic where you could dial one up and dial one down. We would do that at the subconscious level through input through the hypothalamus and the brainstem. Okay, so parasympathetic and sympathetic are innervating by and large the same structures. They're having opposing effects. So here's the last big overview I'll give you, and we'll look back at the notes. Um, Overall, sympathetic is seen as the fight or flight division, that it will turn the structure on, make it go faster. Parasympathetic is viewed as the resting and digesting division, that it's slowing down the structure, that it's turning it off, so to speak. That's a broad generalization, and there are exceptions, big exceptions to what I'm saying. And so that's not always the case, but in general, that's how you can think of the sympathetic and parasympathetic divisions. So going back to the notes, autonomic nervous system is part of the peripheral nervous system, the motor division. It's controlling involuntary movement. So that is in contrast to the neuromuscular junction that we looked at before. And I'm going to skip the pathway for a second. I'll come back to that. So the sympathetic division is the one that we call fight or flight. That's usually going to turn the effector on or speed it up. Sympathetic division is responsible for keeping us in a state of alertness, a state of readiness. And I put some characteristics down: pounding heart, rapid heart beating, increased mental alertness, decreased digestive and urinary function. And if this helps at all, you can think of this as a general redistribution of blood flow. So when we're under overall sympathetic innervation, we're in, fight or flight. Fight or flight means like physically you have to fight or you have to run. And so we perfuse our skeletal muscles with blood. We perfuse the brain with blood. So we're, mentally alert. We're ready to run or to fight. We're breathing heavily. It's not a good time to be digesting the meal and so on. And so you're literally diverting blood flow from, say, like the skin, the digestive organs, to the muscles and the brain. That's the sympathetic division overall. Parasympathetic division, resting and digesting, is usually, going to turn something down, slow it down. And so parasympathetic division is typically, what we're operating under as far as the balancing act, where we have like warm skin, a calm, a state of calmness, a decreased heart rate, a resting heart rate, normal breathing, where our GI tract is working, we're digesting our food, we're filtering the blood, we're doing all of those things, and we're not diverting our resources to um, to take action in a crisis. Okay, so that's a basic explanation of the difference between the two. And I know I gave you, uh, well, maybe I didn't give you an exception. So here's an exception from this overall description. Uh, the parasympathetic division will actually turn the GI tract on. The parasympathetic division is responsible for urination, so it would be responsible for like urinary bladder muscle contraction. Compared to sympathetic, it doesn't want that to happen. Okay, so parasympathetic does turn some things on, especially if you're thinking about, the organs that it's in charge of. So just to give you that. So there are some anatomic differences too. The effectors, that would be an anatomic difference, right? So we're controlling different things. We talked about that. The pathway is also different. And so I want to take a quick look at that with you. I'm going to jump over to some pictures. And I gave you a copy of this in the notes. It's on the last page, but I'm going to look at it from our book here. It's in color. So here, what we're looking at is some of the autonomic differences between autonomic nervous system and somatic. And so here's a somatic nervous system. This is what we talked about, the neuromuscular junction. Now notice this is the central nervous system that they're showing um, in this brown shading. So all of these neuron cell bodies are in the gray matter of the central nervous system. And then what would happen is these axons are exiting through nerves, right, through cranial nerves and through spinal nerves. And so with the neuromuscular junction, what you have is you have neurons that are originating in the central nervous system, one long axon that goes to the skeletal muscle that's being innervated. Acetylcholine is always the neurotransmitter at the neuromuscular junction. It always has a stimulatory effect, right? It's always going to stimulate the muscle to contract. And that's what we've looked at so far. Here, just to contrast this, when you look at the autonomic nervous system, whether it's the parasympathetic or the sympathetic, notice there's a chain of two neurons. And so you're going to have a neuron that exits, and then you're going to have a second neuron that goes to the effector. Neuron exits the CNS, synapse is somewhere in the body. Well, actually, that's not a good example. And then a second neuron goes to the effector. Actually, this example that I just skipped over, neuron going to the adrenal gland, this is an example of the nervous system controlling an endocrine gland, controlling release of a hormone into the bloodstream. And so we talk about this example, the specific example in AP2, and I'm therefore not going to cover it at all. And I'm going to keep it to the straight contrast between the parasympathetic 2 neuron chain and the sympathetic 2 neuron chain. So when the first neuron synapses with the second neuron, you might recall that we said that this would be the presynaptic neuron and this is the postsynaptic neuron, right? And we'd be looking at the synapse. Well, what happens here is because this is going to happen with many, many neurons, not just one neuron will be in this pathway, is that you're going to get a collection of neuron cell bodies out here. And when you have a collection of neuron cell bodies, it creates that bump, like the dorsal root ganglion that we saw. In other words, what's going to happen is where the first neuron synapses with the second neuron, that synapse will occur in a ganglion. Okay, it's going to be that group of neuron cell bodies. And because of that, the terminology here is a little bit different. We would call this first neuron the preganglionic neuron instead of presynaptic, although it is presynaptic. It's known as the preganglionic. And the second neuron, instead of saying postsynaptic, we say postganglionic. And that's the terms that I'm using in the notes, so I just wanted to make sure that makes sense to everyone. Okay, so where our first neuron in the path synapses, it's going to be in a ganglion, so I'd call the first neuron preganglionic, second neuron postganglionic. And that's true with the parasympathetic division, that's the one I just marked up, and the sympathetic division. So with the sympathetic division, you're going to have your preganglionic neuron, it's going to synapse within a ganglion, and then your postganglionic neuron is going to go out to the effector and stimulate the effector. Okay, and so the anatomic pathway is much different from the neuromuscular junction. So I hope that makes sense. The other thing that's very different anatomically is where these preganglionic neurons exit the central nervous system. So I'm going to erase my markup here and go back to the notes. Okay, so going back to the notes, you can see this is where I wrote the pathway is going to be a chain of two neurons. There's going to be a preganglionic neuron that exits the central nervous system. It's going to synapse with a postganglionic neuron within the ganglion, which is the cluster of neuron cell bodies outside of the central nervous system. Now, if we had a cluster of neuron, this is just a little review. If we had a cluster of neuron cell bodies inside the CNS, do you remember what that's called? It's called a nucleus, like the basal nuclei, as an example. Okay, so we can have clusters of neuron cell bodies inside the CNS and outside the CNS. Outside the CNS, that's when we call it a ganglion. So, so I talk about that here at the bottom of page 10, plus I'm talking about where those fibers are exiting the cord. And I'm going to jump to some pictures. So I'll just mention here, and I'll show you this, that there are short preganglionic fibers in the sympathetic division, and all of those arise from the thoracic and the lumbar regions of the central nervous system. Compare that to the parasympathetic division. The preganglionic fibers are long. They go almost all the way to the effector organ. They synapse in a ganglion and send the short postganglionic fiber into the effector, and all of those long preganglionic fibers exit the central nervous system through cranial nerves and in the sacral region. And so let's take a look at a picture of that. So with the sympathetic division, this is the picture from the book. What we want to notice here is that the preganglionic fibers are exiting the cord at only the thoracic and the lumbar regions. That these preganglionic fibers that exit in those regions, which, by the way, if you remember the lateral gray horns within the cross section of the spinal cord are made up of the neuron cell bodies of these preganglionic sympathetic neurons. So these preganglionic sympathetic neurons exit, pardon me, through the thoracic and the lumbar regions, and they enter into what's known as a chain ganglion, which is this thing. And it's what this is a bunch of ganglia that occur at the same level, basically. And because they occur at the same level, they fuse together. And when the preganglionic fiber, this preganglionic sympathetic fiber enters in to this ganglion, this is where it usually synapses with the postganglionic fiber. And so therefore, what happens is your preganglionic fiber, it's pretty short because this ganglion is very close to the spinal cord. I'm going to show you another picture in a second. And then your postganglionic fiber would be much longer, going all the way out to the effector. So that's the basic setup of the sympathetic nervous system. So just to take a look at a few more pictures. Here is a diagram of the chain ganglion that I was just referring to. You can see it's right by the vertebral column. And you can see when a spinal nerve exits. So there we have ventral root, dorsal root, spinal nerve. There are going to be communicating rami that bring the neurons from the autonomic, from the sympathetic division into this chain ganglion. And from that point, what happens is, like I said, is they go into the chain ganglion, they synapse, and then a long postganglionic fiber goes out. That's very typical. But there are other possibilities. Here's another possibility. It could travel within the chain ganglia, and so it could come into the cord at one level, ascend or descend within the chain, and then synapse at a different level. So actually, here we have it coming into the cord, ascending, and then synapsing at a higher level than it entered the cord. And then another possibility is some preganglionic sympathetic fibers enter into the paravertebral ganglia and they don't synapse. They leave. They synapse in another ganglia. It's called a collateral ganglion. And the cells that do that are going to be innervating the GI tract, and these are called the splanchnic nerves. But just to summarize, the short preganglionic fibers that enter into the paravertebral ganglion, they can synapse at that level, they can synapse at a different level, or they might pass through and synapse in a different place, and those are the splanchnic nerves that are innervating the gastrointestinal tract. So taking a look at our notes, the sympathetic division is known as the thoracolumbar division. That's because all the preganglionic fibers exit at that level. All the preganglionic fibers are short. They enter the paravertebral ganglion, okay, and then they synapse at that same level or different level, usually, except for the splanchnic nerves, and then you'd have a long postganglionic fiber go all the way to the effector. So that's the basic anatomy of the sympathetic division. So let's take a look at the parasympathetic division. So parasympathetic division is known as the craniosacral division. So the parasympathetic division, as we said before, it's innervating, the same effectors basically, except here all of the preganglionic fibers are exiting through cranial nerves and exiting through the sacral level of the spinal cord. So sympathetic preganglionic fibers are thoracolumbar, parasympathetic cranial, sacral. They have long preganglionic fibers, so there's no paravertebral ganglion, in other words. They go almost right out to where they're going to have their effect. They synapse right at that level, right where they're going at the effector in a ganglion. And overall, we can call these ganglia terminal ganglia because it's at the terminal end of the pathway. They each do have their own individual names, and you don't have to worry about that at all. But they're going to synapse in a terminal ganglion, send a short postganglionic fiber into the effector, and typically that postganglionic fiber will release neurotransmitter and have an effect on, a wide, like a collection of cells right within the effector. Okay, so that's how the parasympathetic division looks anatomically. Pardon me. So parasympathetic division, it's known as the craniosacral division. The preganglionic fibers come from those areas. They're long. They go almost all the way to the effector. They synapse right outside of the effector in a terminal ganglion, and then they have a short postganglionic fiber that goes into the ganglion. And so to go back to the picture that we were looking at the start of this little lecture, the one that I gave you in your notes on the last page, if you wanted to mark that up at all here. Let's just take a look at what's happening, and we'll go a little further too. So in the sympathetic division, what we're saying is that there's going to be a short preganglionic fiber, that this fiber hardly makes it, right outside of the vertebral column, and already it's in this ganglion. This is what you call the paravertebral ganglion. The neurotransmitter that's used by the preganglionic fiber is acetylcholine, and it always has an excitatory effect at this point. You're always going to excite the second postganglionic neuron. So then we're going to excite the long postganglionic neuron, and that fiber is going to go out to the autonomic effector, where it can have either a stimulatory or an inhibitory effect. I'm going to talk about that more in a minute. In the parasympathetic division, what we see is the neurons exit through the cranial nerves and the sacral nerves. They go almost all the way to the effector. This would be a terminal ganglion. They're going to release the neurotransmitter acetylcholine. It's going to synapse with the postganglionic neuron, which is a short neuron that goes right into the effector. And again, it's going to have either an excitatory or an inhibitory effect. So the preganglionic neuron in the parasympathetic division is the one that's very long. Pardon me, sorry about that. Synapses in the terminal ganglion, and then the short postganglionic fiber is going to go right into the effector. And so take a look at the neurotransmitter situation here. Notice that acetylcholine is used in the autonomic nervous system by all preganglionic fibers, whether it's sympathetic or parasympathetic. It will always have an excitatory effect. Once you get into the postganglionic fiber, here we're seeing a difference in the neurotransmitter that's being used. In the parasympathetic division, the neurotransmitter that's used is acetylcholine. And I'm using purple because that acetylcholine could have an inhibitory or an excitatory effect. It would depend on the type of receptor that binds the acetylcholine. So keep in mind from Unit 5, the effect that's felt at the postsynaptic neuron, the postsynaptic potential or the graded potential, depends on what type of ion channel you're opening. So these receptors are chemically gated ion channels. So this could be causing a depolarization or a hyperpolarization in the effector, and that's what makes it excitatory or inhibitory. You're not going to have to worry about specific examples, but I do in the notes tell you about different types of acetylcholine receptors. There are two main categories. They're called nicotinic and muscarinic, both named after a plant toxin, nicotine, and then a type of mushroom that binds to these receptors. And so there are main receptor types that will dictate basically what type of effect this is going to have. Now, if we look at the sympathetic postganglionic fiber, all of those fibers release a different type of neurotransmitter called norepinephrine. And norepinephrine will also have either a excitatory or inhibitory effect at the effector. Again, it depends on the type of ion channel that's being opened. So there are different types of norepinephrine receptors. They're called alpha and beta. Overall, alpha is typically excitatory, beta is typically inhibitory. But even from there, they're subcategorized. So there's alpha 1, alpha 2, beta 1, beta 2. Same thing with the nicotinic and muscarinic. And so this gets beyond the scope of our course, except I just want you to know that the effect, whether it's going to be excitatory or inhibitory, it doesn't depend on the neurotransmitter. It depends on the type of receptor that's binding the neurotransmitter, because that's going to be the ion channel that's causing the hyperpolarization or the depolarization, making it excitatory or inhibitory. So let's jump back to the notes. And I wrote all of this down for you. So I made a little chart so you could see that the neurotransmitter used by all preganglionic fibers is acetylcholine, and also by the postganglionic parasympathetic fibers, norepinephrine is used by the sympathetic postganglionic fibers. So acetylcholine, that is released by, most of these fibers. Those neurons are known as cholinergic fibers. Okay, so a cholinergic fiber just means a neuron that's going to release acetylcholine as its neurotransmitter. And there are two major types of cholinergic receptors, nicotinic and muscarinic, as I mentioned. Norepinephrine, fibers that release norepinephrine are known as adrenergic fibers. And there are two main types of adrenergic receptors, alpha and beta. Both of these, again, subcategorized. And so these receptors are really determining, the effect that we see on the affect the postsynaptic potential. So the effect, whether it's excitatory or inhibitory, it's going to depend on the type of receptor, not the type of neurotransmitter. Now, keep in mind, I did say, and this is true, this is a fact, that the preganglionic neurons that release acetylcholine, they do always have an excitatory effect. It's at the postganglionic fiber where there's more question mark. So neurotransmitter receptor is a chemically gated ion channel, so the effect depends on the type of channel that's opened, right? Did a sodium channel open? Did a potassium channel open? And I'm even simplifying things here. It could be something different, but again, it goes beyond the scope of what we're doing. So I hope that this overview of the autonomic nervous system makes sense, helps out, and I will talk to you again soon. Thank you.